Perfect. We have entered the live session and we are also recording this session so that it can be shared after the conference with everybody.
Samir is with us.
Good afternoon, everybody. Just to advise that and welcome you to the afternoon session. The session will commence in 3 minutes. Thank you very much.
Eunice, I start sharing my screen if it is okay. My only concern is that.
Yes, they'll allow me just to introduce you and then
Sure.
switch over to you and then you can do the screen share. I don't know, you can probably start immediately with that if you like, but
I prefer to share it in advance just in case that it doesn't want to share the correct screen. Okay, I share it now.
okay, I see your background, yes.
And now you should be able to see the presentation.
Yes, I see in a web browser Chrome, yes.
Yeah, but unfortunately, when I want to do that offline, it goes to the main screen. It doesn't show the offline version. There's a reason that I'm concerned to make sure that I'm sharing something that is usable for audience.
Okay. You said 5 minutes for the Q&A.
Yeah, I can finish around 20 past 2. It depends. I can speak for hours, but let's see how fast you can.
Yeah, depends. Hopefully we get, people don't have internet problems and we do get questions. So I don't want to stop and then you still have things to say and then we don't have any questions coming.
So I think that I finished 20 past 2, between 20 and 25 past 2, easily.
You may have to go on until 2:30, so just know about that. Okay.
Slowly.
Right, you're nearly there.
Good afternoon, everybody. Welcome to the afternoon session. I would like to hand you over to our chair for the afternoon session, who is Mr. Ines Skeppers. Mr. Skeppers, you are welcome to open the session. Thank you very much.
Thank you, Deirdre. Good afternoon, everybody. I'm Ines Skeppers from CHPC, and I'll be chairing the first session this afternoon. Our first speaker will be Dr. Arslan Imamekoi from University of Johannesburg. He's a postdoctorate researcher in the Center for Ecological Genomics and Wildlife Conservation at the University of Johannesburg under Professor Teske. And he uses genome-wide data to study marine animals. The title of the talk is High Performance Computing in the Era of Genomics. Thank you, Dr. Arslan.
Thank you. Afternoon, everyone. I hope that you are hearing me. Okay, I start now. Wildlife genomics, probably you think that we are doing or creating something like this, or this, or maybe this, or this in our lab, which is not the case. Actually, we study the DNA. And you probably remember this 2-3 meter long string of DNA that is tiny compared to the hair's diameters, and the combined length of the DNA inside the cells is twice the diameter of the solar system. What happens in genetics is that in 2017, we witnessed a revolution in genetics. That revolution was started when, instead of sequencing, that is deciphering the code or identifying the homopolymers of the DNA in one single strand, which we used to do for 30, 40 years, we started massively sequencing multiple million, if not billions of strands at the same time. And it gives rise to the new school of technology that we call it massively parallel sequencing. That means that it's a traditional sequencing on steroids. We parallelly sequence multiple strands of DNA, and we need to decipher the data from in such a large scale. Now we have the issue of big data, as you see. We are overwhelmed with the amount of data that this new method can extract from billions of strands of DNA. It has its advantages and its disadvantages. The advantage is that the cost has not decreased dramatically. For the Human Genome Project, we had to invest $3 billion over 1 decade to have the first human genome ready. But these days, we can invest less than $1,000 and less than 24 hours to have a whole genome for the human sequence. But the bottleneck is that amount of data is overwhelming. And computational resources and the skills that are needed to analyze this data is really extensive. And the big issue is that traditional biologists are not typically trained to deal with such a scale of data. And that constitutes a major bottlenecks in analyzing the data. I just narrate a quote from Professor Richard Dawkins that he mentioned that modern genomics DNA is nothing but a very long string of data and needs to be dealt in the same way that we used to deal with long data in computer science. Change from genetic to genomics wasn't a transition. It was a complete transformation of the field that we need to take into consideration when we want to analyze the data. At the end, all these technologies are supposed to help us to extract more data and interpret it. Sequencing millions or billions of DNA strands in parallel per cell is not something that can be meaningful. Just to give you an idea, this is what we used to do in genetics. It was just a one gene, one precise position in the genome, but no, we need to deal with something like that. It's all the chromosomes in the typical mammalian genomes. And we need to decipher the information from such an entangled web of the chromosomes in at least 3D dimension that we can see and probably more dimension that we cannot see. From now on, I walk you through some of the basic workflow that we follow in our labs and see how CHPC is crucial for analysis and how we depend on CHPC for producing scientific outputs. The first application of the CHPC in our genomics lab is assembling full or partial genome. Let's start with that one. You remember that one? This was the whole chromosome in the typical mammalian genomes. Now we need to sequence it. What we will do is we need to chop it to the pieces that a sequencer can actually analyze them. We call it shotgun sequencing because what we do is we actually explode the DNA in small pieces and start rebuilding, reconstructing the whole massive strand by overlapping segment of DNA. As you see, we stitch one end of one to the beginning of the others, and as such, we continue until whole genome, but mainly part of genome, are successfully assembled. Later on, based on some conserved motifs in all the eukaryote or all advanced animal genome, we start predicting the position of the genes on the de novo assembled strings and try to see, based on the homology to the known animals, mainly model animals like humans or rats, we try to infer the function and predict the function that they might perform in the living cells. As such, many of you that are familiar with computer science, you suddenly realize that from here onward, this is not genomics anymore, it's your string manipulation in computer science. We have hidden Markovian chain, we have complex indexing algorithm, we have de Bruijn graph to assemble the shotgun sequences based on overlaps. And it's an old history that we need computational resources to perform that. It is not something that one can perform on a single computer or in the normal cluster of few computers that we usually have in our universities. Modern indexing algorithm, complex machine learning algorithm that is required to predict the function of the genes and require large memory computational resources, vault time that are usually crazy. And as you see that from now on is the overlap between computer science and modern genomics. And as we mentioned earlier, it is something that biologists are not used to that in such a scale. The main focus of our lab is evolutionary genomics. It looks a bit complex, but I try to explain you in an easy way what is evolutionary genomics and what we perform in our lab. It's just predicting how the DNA variation in population change backward and forward through the time, or have changed backward or will change forward through the time. Just we start with this animation, which is the old Beast and Beauty story. Don't be surprised, Beast and Beauty are core packages in evolutionary genomics. It's not just coincidence that we have these two figures next to each other. Okay, these two fall in love and now they marry, and now it's time to offspring. And we know that offspring can be everything between a mom genetic makeups and a dad genetic makeups. And okay, after that, these offspring start mating among themselves. That means that we have more and more different forms and morphologies that are not observed in the parents, but are new to our population. And this is a never-ending story as far as they can reproduce. And they keep producing offspring that inherit part of genomes, part of the traits from the parents, and part are completely new, have never been observed in nature. And this continue and continue and continue. We'll have more morphs, more traits that are somewhere between the parents, but are not identical. And at the end, we get overwhelmed with number of shapes and function and traits that we observe in the wild populations. It's not surprising given that we know that we all started our life as a bacteria a few billion years ago. What will happen is that you see that, okay, from that two initial traits, we have the jungles of different traits. Some of them has nothing to do compared to their parents, but we still need to be able to trace them back to their biological parents or evolutionary parents. Okay, it was an animation, but I wanted to explain to you how it actually, how can it be performed in real world situation. Just look at this animation, and before I start, I wanted to tell you that just treat the green dot as beast and blue dot as beauty. Okay, now they mate and offspring, generation after generation after generation after generation. They inherit some of the traits from their parents and some new traits that are completely de novo, means that they didn't exist in the nature. Repeat it again and again and again. I think that for you with background in computational science, you saw you easily notice that it's not something that we can do in a serial manner. We need to do that simulation for at least hundreds of millions, not billions of times. The reason is that we cannot really predict the future. We just want to integrate over all the outcome that actually these two parents might give rise to. This is a reason that we need to run them in parallel. And parallel means that multiple codes. We need to write a script that can be easily scaled up using at least different codes. We hope that MPI works, but in genomics, we are still far behind engineering science. Most of our MPI scripts are not as advanced as engineering scripts, but this is where actually the resources start being crucial for us to analyze. Just to give you a real application of that in real world, my lab actually working on one of the most endangered fish on the planet. We call it Australian pipefish. Eastern Cape is home to few pockets of this animal in a small number, and they are classified as critically endangered. The question is that my lab is now involved in translocating some of them from the area that they actually still exist to the area that they have been wiped out due to unknown reason, maybe a combination of the contamination, habitat degradation, or God knows what. We need to select those individuals that needs to be translocated, that is moved from one location to other carefully. In terms of genetic variation, we want to make sure that the population after 100 generation, 200 generation, we still have enough variation to withstand the wear and tear of time. But it's not possible if you just predict based on two individuals. We need to move to perform multiple rounds of the simulation and see what is the outcome of translocation after 100 generation, 200 generation, or 300 generation or so on. And this is where we actually need extensive computational resources to be able to do that. And our decision and the robustness and accuracy of our simulation is the difference between survival and extinction for these animals. It's not something we can do in serial manner. It needs to be parallelized, and it needs to be done in massive scale. We need to take the slightest probability into consideration. Okay. The last century, we witnessed one of the biggest revolutions in genetics. They are these inspirational old guys that explore the boundaries of the science of the genomics. But we need to accept that science has changed. The amount of expertise that is needed to start a genomic project from field up to the publication-ready data analysis and scientific output is usually beyond a single lab or even a single university. The future of the science in genomics is based on collaboration. Scientists need to work as a team in collaborative units, and the collaboration is critical if we want to stand a chance to explore the new frontier that the genomic era opened to all of us. We miss these guys when they personalities, hard working in the lab, but we need to be ready for the new era in genomics. I just want to conclude with few marks is that in wildlife genomics in South Africa, having access to HPC resources is fundamental. Sadly, we should tell you that it is black and white. That difference is that with HPC resources, we can produce output. Without HPC resources, we cannot. There is no gray tone in between, at least as yet. And it is a turning point in wildlife studies in South Africa. We can swing either way. We can return to a comfort zone in traditional genetics. It's fantastic. They have done heaps of interesting results. We respect all of them. But if you want South Africa to be a leading country in wildlife genomics, needed to get ready and facilitate this transition into new area in life science. And for that, having access to massive HPC resources is crucial. At the moment that we are speaking, there is no reliable alternative in South Africa. Our research budget is so small that using commercial service provider like Amazon, Google, Compute Engine, others is not feasible. We cannot run a Compute Engine in Google for something like 20 days. It's more than the whole budget of the university for a year. And just to conclude, I wanted to tell you that don't think that wildlife genomics is something related to wildlife. Wildlife genomics is a proxy that we can study human health and human well-being and future of the society. Wildlife, very important and valuable preserve, they give us more flexibility to study the biological and evolutionary patterns. No, we are in the scale that we can not only study the behavior, we can study the pattern in the large scale. But the same patterns, these core patterns, are very conserved across the domain of life. It means that each new wildlife species we identify, each species that will be saved from extinction, might help us in developing new antibiotics, anti-inflammatory, anti-cancer drugs. And if you just survey the pharmaceutical studies, you see that how many of our drugs actually comes from animals. And sadly, many of them disappear soon before even being recorded. And definitely the solution of many of our health issues are already been answered by one of these animals that's waiting for us to be documented, to be discovered. And I think that I'm reaching the end of my time. Thank you, everyone. And I want to reiterate that don't think that in wildlife genomics, we create mutant animal like that. We don't do that, we study DNA. Thank you. I'll give the virtual podium back to Ines and waiting to answer your questions.
Thank you, Arsalan. I would like to ask if there are any questions. We haven't seen any in the Q&A panel. So if you have any questions for Dr. Emamia Kwe, please let us have them.
Seems that no one is interested about mutant animal in this. Okay, thank you, Reynold, Dr. Quinn Reinhardt. Ines, can I start answering the question? It seems that I have lost Ines. Are you hearing me?
Yes, maybe you can just tell us how the CHPC has been useful to you.
Yeah, I completely got, that's all right. I just got something in my chat box from Quinn Reynolds, Dr. Quinn Reynolds. Yeah, you're absolutely correct. What we will do is that for assembly, we definitely use a large nodes. We need a memory in terms of the RAM. 1 terabyte is great, but still it has been cases that we had to ask for extension and we need to chop our data to smaller pieces. I can tell you that as a rule of thumb, for all of our assemblies, we have a difficult decision. We have difficult choice between resources in terms of CPUs and resources in terms of memories. So far in our lab for assembly, we go for the large big mem or large memory queues. But for simulation, the way to go is for large cubes that we can parallelize multiple simulation in multiple nodes. So far, we used our normal SMP nodes. If we get sure that we can scale it up on MPI level, we use large node, and few programs actually has been scaled up very well. We scale it up to all the available large nodes, but yeah, at this stage, genomic science is lagging behind engineering, and MPI is still in its infancy in genomics, but I wish that we could have scale up everything into MPI level. Okay. Any other question, critiques, suggestions?
There's a question from Dr. Govinder, and he's asking whether it's possible to port any of the simulations to the GPU.
Yes, at the moment, our famous Beast and Beauty that I show you, actually there are two main packages in evolutionary genomics that are widely used. They scale up relatively well in GPUs. Sadly, for many others that have few Python codes, I personally didn't have much luck, but I know that some of them can scale up relatively well. But let's first optimize it for CPUs and reserve the GPUs for the next generation of the genomics, computational genomics that comes. Some of them, yes, just to answer your question, but majority, no. We had to stick with nodes with more computational resources within a single node, and maybe MPI at the next level.
Thank you. We have another question from Rafael Ployer.
Sure.
You mentioned that biologists are not trained for handling this large amount of genomic data. What is your opinion on, and what should we do to improve our capacity to store and share these large amounts of data?
Actually, I don't mention biologists. I say that traditional biology. Traditional biology used to work with one genes. Probably you remember that just sequencing half of the insulin gene was something that would have given you, something very close to the Nobel Prize. But these days, sequencing the whole human genome, you cannot even publish it anywhere. It's such a normal and basic things. The reason is that, as I think that we need to start a little bit, invest on computational part of the biology, which was completely overlooked so far, and the reason was understandable. Many of us start biology because we were not good at math and statistics. And at the end, when we face up with so much amount of information and genomics data, we have to go and ask computer scientists to come and treat them, as Professor Dawkins mentioned, as a long string of data. And probably my generation, we all remember the first time that we sit with computer scientists and they all tell us, look, don't speak about DNA, just say that, okay, this is a string and you want to do that and that. That string can be anything. Of course, there are biologists that are very good at computational science. Majority of computational genomicists these days are either biologists or biologists that later on train and learn the computational part of the biology. I think that we definitely need to invest more in computational part from high schools and just make our students brain or view about the genetics that it has changed. It's not what it used to be. Watson, Crick's, all of them are very nice person, but I'm sure that if they were alive today, they would recommend you that, look, biology has changed. Either adapt or die. We can swing back to where we used to be, but that we might be able to do some good science, but we might not be able to keep our competitive edge. And as I mentioned at the end of the slide, I think that the first step for us as established researchers is an interdisciplinary collaboration with scientists from different fields, computer science, statistics, biologists, and or integrated knowledge will be embedded in something that is competitive in international arena. At this stage, even in big countries, even in more advanced country, you will see that that amount of expertise is not available in one single lab or in one single research group. Investing in computational biology, changing our students' view of the biology, that modern genomics is nothing but dealing with big data. It is nothing but that. If you think otherwise, it is not genomics. And at the end, multidisciplinary collaboration in collaborative units that we can learn and we can share our skill together. I think that this is my opinion, and as you see, that is the opinion of all the big experts in the field, that collaboration, and interdisciplinary exchange of ideas. Like in CERN, if you see it in physics in CERN, they are guys that develop the CERN, they are the guys that doing the experiments, they are the guys that keep this whole system working. They are theoretical physicists that actually make sense of those data. And they all work in very close collaboration. Sadly, and this is a reality, we are lagging behind physics and engineering in that field. We need to catch up if we want to stand a chance to be competitive. Yeah, this is my question.
Thank you, Arslan.
Pleasure. Hope that you have enjoyed it.
Okay, I don't see any other questions coming in. I want to like to thank you very much for your interesting talk.
Pleasure. I hope that you have enjoyed it. And yeah, I just wanted
And
to talk to that serve as a place to share opinions and see how important it is that we collaborate together and how crucial CHPC is for all of us.
Thank you. Thank you for taking part in our first virtual conference.
Pleasure, Ines.
Okay, so our next speaker will be in 2 minutes' time, so we will keep this open and wait for other people who want to coming in to join.
Sure.
Got a bit of a cement truck outside, concrete truck.
I've shut all my doors and windows and ordered everybody to stop walking around in the corridor outside.
The walls are literally vibrating, yeah. Okay, should we start? Hello?
Oh, hello. Sorry, I thought you were going to introduce me. Don't worry, I'll do it myself.
I'm not sure whether I'm live yet. Deirdre, can you confirm?
Hi, Ines. We have got 32 participants already that have entered. We're welcome to start. Dr. Reynolds has already shared his screen with us. So please go ahead and introduce Dr. Reynolds.
Good afternoon, everybody, to this session of HPC applications channel. Today, our next speaker is Dr. Quinn Reynolds of the Pyrometallurgy division of Mintech. And the talk will be toward the efficient, scalable, computational fluid dynamics solver for mesoscale phase separation problems. Thank you, Quinn.
Okay, thanks, Ines. Good afternoon, everybody. I'm just going to stop my video because my internet connection is not that fantastic. Hopefully you can still see my presentation okay. So, yeah, today I'm going to be talking to you a little bit about some work that we've been doing in the last year or two, developing a new computational method for phase mixing and separation problems. Hope you've all been enjoying this new normal, the online conference format this year. It's certainly a little bit different compared to past events. But the one thing I do like is that you can wander off for a cup of coffee whenever you want. So if my talk is a bit boring, please feel free to go ahead and indulge your caffeine fix. I won't judge. So I work at Mintech, which is one of the South African government science councils like CSIR. We do R&D mainly in the fields of mineral processing and metallurgical engineering. Just a little bit of background about the place. We're not very big by comparison to other institutes. We're only about 500 people. We're all based in one location in suburban Joburg. But we do have a few interesting and unique features. In particular, we're probably the only place in the world that you can go and get access to expert R&D skills and pilot scale testing pretty much across the entire minerals beneficiation value chain, all the way from run of mine through to final products and all under one roof. So the department I work in is called Pyrometallurgy, which is also the technical discipline that we work in. In a nutshell, it's the art and science of building and running furnaces in order to convert raw materials into products of value. So if you enjoy setting things on fire and melting them, then this is probably the job for you. So in Pyro, we really do have some of the most challenging and intractable engineering problems in the world. In particular, we've got a lot of multi-phase flow problems. A very common one we run into all the time is mixing between molten slag, which is an oxide-rich phase, it's a bit like lava, and molten metal, which is usually your valuable product that's coming out of the furnace. So slag and metal behave a little bit like oil and water once they're in the liquid state at high temperatures. They don't like to mix together, and they usually form separate layers inside your furnace. So, of course, you've got to get those molten materials out of your furnace at some point. That's usually done by a process called tapping, which involves opening a special channel in the wall of the vessel and allowing the contents to drain out under gravity. The problem with tapping is that it's a very high energy multi-phase flow, and it can cause a lot of mixing of one phase into the other. And there are quite a few other processes in furnace technology that cause similar effects, and the end result is always the same. You can end up with almost an emulsion of small metal droplets stuck in your slag phase. And the problem with that is that once your phases are mixed, it can be really difficult to unmix them again. So usually your metal is your primary product of value, and you don't want to be throwing away lots of that in your slag after it solidifies. These molten materials have some quite unusual properties. They've got very high surface tensions and very large differences between phase viscosities and densities. And all of those can affect the settling and separation, sometimes quite negatively. So we're showing some examples here of metal droplets collecting in the bottom of furnace tapping ladles and crucibles this is from some tests that were done by colleagues of ours in Norway a few years ago. And it just illustrates the problem. This is material that was probably sitting in a ladle for a few hours and it hasn't actually separated. So if we want to model these kinds of problems, well, there's a few different ways we can go. We can use mixed or meshless methods to treat the dispersed phase droplets as discrete particles, or we can forget about resolving the droplets at all and just treat them as a continuum which can intermix with the slag. Oh, by the way, I'm talking mostly about droplets in this presentation, but of course the discussion applies equally to bubbles and any other problem involving one phase dispersed as small particles in the other. So of course the problem with these traditional methods is they need quite a lot of outside help to make them work. Although they're really good for modeling large-scale engineering problems, that really means that by necessity they have to approximate a lot of what's going on down at the scale of the individual droplets. To do that, they have to lump a lot of the droplet interaction physics into empirical models that describe phase mixing and decoupling. And that empirical input needs to come from somewhere. So it's either going to come from experimental data and test work, or it's going to come from other more detailed mesoscale and microscale models of these processes. And that's really what we were interested in here. Methods to resolve not only the droplet motions in the continuum phase, but also their deformation, breakup, and coalescence in large groups, which is the essence of this mesoscale thing that we're talking about here. Mesoscale models can be really useful for fundamental studies and also for numerical experiments to quantify empirical parameters for those engineering models. So one of the ways you can do this is something called the multiple marker method. It's a handy numerical trick that you can use for problems like this. And the idea is pretty simple. You basically treat each droplet as a separate phase in the model. So you can avoid the problem of numerical resolution limits causing premature droplet coalescence, which is what happens in standard VOF and other methods. The big drawback of the standard multiple marker method is that it's only really good for problems with very small numbers of droplets, like pair collisions or something like that. As soon as you go up to even just a few tens of droplets, you have the equivalent number of phases that you need to model mathematically, and the performance of that multi-phase flow algorithm pretty much falls off a cliff then. The other problem with multiple marker methods is they don't handle droplets breaking up or coalescing together very elegantly, if at all. So we came up with an extended version that we call the dynamic multiple marker method, or DMM, to try and address a few of these limitations. The concept is actually pretty simple, although the devil is always in the details. So in DMM, the first step is to use the current phase fields to detect contiguous droplets and assign them unique identifiers, which we're just showing by colors in this picture. So each of those droplets has a unique number that's associated with it. After that, we use the geometry information from the droplets and the phase fields that they're embedded in to determine if the droplets are physically close enough to be in contact with one another. And we build a contact graph structure, so something like this. And then we can use graph theory. So then we can use something like a graph coloring algorithm, for example, to identify a minimal number of marker phases, which ensures that no droplets in contact are sharing the same marker phase. And then after all of that, you reassign the droplets to their new marker phase fields and you carry on with your VOF calculation as normal. So usually the DMM method is used in conjunction with subgrid models, which then describe how the thin film between two droplets evolves after the film thickness drops below the mesh resolution. The nice thing here is that you can plug in kind of whatever you want, everything from empirical drainage models down to fundamental Navier-Stokes models, all sorts of things. So for this work, we opted for a very nice field-based model that was developed by my co-author Oliver Oxleby and one of his PhD students a few years ago. Essentially, it casts the Reynolds film drainage equation into field coordinates and then solves it only in the mesh cells where the films are active. So that is between pairs of droplets that are in contact with each other. You can also model how films rupture using different methods. The simple one is what we used here, which is essentially just to look for a minimum stable film thickness. Basically, once the film gets too thin, it bursts and the two marker phases on either side get reassigned to the same phase so that they can instantly mix together. So with all that, we needed a platform to test the concept out on. Because we're open source people at Mintech, we went for OpenFOAM and our standard kind of fleet of open source support tools. These are nice because they give us all the access to the code that we need to do things like verification as well as extension and modification. And they're generally very performant on the HPC systems that we're interested in using them on. Unfortunately, as it turns out, we did open a bit of a can of worms here. So although OpenFOAM is C-based, it's been so heavily abstracted and templated that it makes it pretty much unrecognizable as standard C++. It's almost like writing code in a peculiar dialect all of its own. And that dialect is really excellent for rapid application development of solvers, which use more standard continuum mechanics formulations to describe them. But in our case, we had these big chunks of the algorithm that were decidedly non-standard. And that meant we really had to abuse quite a lot of OpenFOAM's internal structures to get them to do what we wanted. In particular, getting those non-standard parts to work properly in parallel settings kind of turned out to be very tricky indeed. Probably one of OpenFOAM's greatest strengths is that it abstracts parallelism away very, very efficiently. So as a solver developer, you almost never need to think about it. But again, it only works for continuum mechanics fields and operators. It doesn't really work when you're doing funny discrete math stuff. So we had to write up a lot of that more or less from scratch. And as you might expect, that meant quite a lot of a long and painful process of testing and fixing and debugging and optimizing, at the end of which I don't think any of us ever wanted to hear about OpenFOAM's p-stream classes ever again in our lives. On the plus side, it did also push us to extend the capabilities and functionality of the solver along the way too. And we added quite a bit in terms of new boundary conditions and CFD physics to handle increasingly complicated test cases. A lot of the major head scratching really happened around the parallel performance optimization though. And it's important here to point out that a lot of the problems we encountered only popped up when we started pushing the solver to each successive new level and scale. A lot of my sloppy parallel programming really got shown up badly every time we scaled from 10 droplets up to 100, and then we'd get all the bugs fixed out, then we'd go to 1,000 and there'd be more bugs and so on. So I've always been a very big advocate of testing your code very thoroughly before you get anywhere near deploying it on large systems like CHPC Lengau. But in this case, it was really the sheer scale of the facility and the size of the problems that we could run on it that were what led us to a lot of the bottlenecks. And it's still sending us back to the drawing board even today. Okay, so let's have a look at some results from all of these optimizations. The first pass at the DMM solver that we came up with about 2 years ago already scales really well with droplet count, as you can see here. This is just for a very simple 2D test case. By the time we get up to a few tens of discrete droplets, the DMM method is already running about 2 orders of magnitude faster than the standard method. So we were pretty happy with that. Of course, then we got cocky and we tried it out on bigger problems and we found that it fell over and we had to go back and fix it and wash, rinse, repeat for several iterations of that, which is pretty much why I don't have any hair anymore these days. But on the plus side, it does make the current solver even more performant. So with all the newer optimizations in play, we more than doubled the performance of the old code. And that improvement only gets bigger as the droplet count rises. And remember, thank you to CHPC for getting us here. We would never have done these optimizations otherwise. So we've also started doing some more traditional benchmarking of the DMM solver, looking at parallel scaling mainly. In this example, we had a simple settling problem with around 1,500 droplets present. The solver doesn't do too badly here. We get nice linear scaling up to about 100 cores or so, which in this case equates to about 20,000 mesh cells per core. That's in the vicinity of what we expect from standard OpenFOAM CFD solvers. And we do still have quite a few tricks up our sleeve, which we hope can help to improve the parallel efficiency as well as the absolute performance of the DMM algorithm. So kind of watch this space and we'll see how far we can go. Okay, so to test out the DMM solver, we started with arguably the simplest possible settling and coalescence problem. So that's just a single droplet falling down into a quiescent pool. The problem with doing experiments on slag metal systems in the molten state is that if you want to use the real materials, they are at crazy high temperatures, typically 1,500 degrees or so. And that is really, really difficult and dangerous stuff to work with. So what do? We go looking for room temperature materials that have similar surface tension and continuum phase viscosity and phase density differences and things like that. But it turns out there's almost nothing that matches perfectly. We used mercury and silicon oils in this particular experiment, but it's not a perfect analog, as you can see. So the main aims of this exercise were to see if we were getting realistic qualitative behavior in the model, and also to see if it was possible to estimate a value for the critical film thickness that triggers film rupture. So here we go with an example of the experimental mercury droplet test on the left and a computational model of the same test on the right. The tests were all filmed with high-speed video cameras so that we could directly observe the droplet deformations and phase interfaces during impact and rupture. You can see we're not getting it completely right. So there's some initial oscillations and surface vibrations occurring on the experimental droplet after it's released that we can't really duplicate in a computational model, and the timing doesn't match perfectly. But the overall qualitative behavior is quite similar. The droplet lands on the surface of the pool, it rebounds a little bit while the film drains, and then it collapses as the film ruptures. Okay, so basically what we're showing here is the results from computational models of each experimental test that we did with that high viscosity continuum phase. So what you're looking at is the evolution of the thickness of the film between the droplets and the pool in time after the point of impact, which we're getting from the Reynolds equation subgrid model in each case. So in general, as the droplet approaches the pool, there's a steady drop in film thickness, followed by lots of fluctuations after it impacts. And that happens because as the droplet rebounds off the pool surface, it creates an oscillating positive and negative pressure that basically sucks in and spits out fluid out of the film. So based on when film rupture and droplet coalescence happened in the experiments, we were able to estimate a value for the critical film thickness. It's quite tricky because the dips and oscillations make it hard to determine a consistent value of H crit that matches all the experiments perfectly. That really indicates that we need to do a bit more work in this area. We're already busy looking at improvements to the film submodel and rupture criteria that should help us address this once we get the kinks ironed out. Still though, if you have a look at a couple of particular cases, you can see that we're getting roughly the right behavior going on. Fall height in particular has a consistent influence on the film behavior relative to the critical film thickness. When we release the droplet from higher up, we get deeper initial drops in the film thickness before the rebound. And vice versa, when we release the droplet from closer to the pool, we get a shallower drop. This is really about impact speed. So the faster the droplet hits the pool surface, the more likely the film between them will thin fast enough to rupture almost immediately. So if the droplet survives that initial impact, it could then last quite a bit longer at the pool surface before coalescing. And that matches quite closely to what we saw in the experiments too. So after looking at single droplets, we started to move into what DMM is really designed for, which is problems with lots of droplets. This was a comparison that we did looking at the average settling velocities at different droplet concentrations. So all the way from very dilute concentrations like the picture on the far left there up to denser configurations like the one next to it. This is a problem called hindered settling, and it's been very well studied in hydrology and chemical engineering. There's some good empirical correlations that were originally developed by Richardson and Zaki in the 1950s, and we were quite curious to see if the DMM model would even work here. Remember, DMM is essentially a full CFD solution of the flow inside and outside all of those separate particles. It's not a particle-based method at all. And it turned out that we did actually get quite good agreement, even in this very simplified case. So that was pretty cool to see. Okay, so moving on from the validation, we decided to take a look at a simple representative slag metal settling problem to get a bit of an idea of how it would react to variations in the parameters. We set up a case using these parameters shown here, which are pretty typical for the kind of ferroalloy smelting processes that we have a lot of in South Africa. And we basically filled a small region, a few centimeters in size, with a random collection of metal droplets to see how they would settle out. Right, so here are some simulation results. The first set of tests looked at changing the slag viscosity while keeping everything else the same. In the animations on the right there, you're looking at the metal phase shown in gray, and the slag phase in between is colored by the velocity field. You can see a noticeable decrease in the settling speeds as we'd expect as viscosity increases, although the rates of coalescence at the pool surface look like they're quite similar. As a crude indicator of how fast the phase separation is happening, we also track the number of droplets in each model case over time, which is shown in the graph on the left there. There's definitely quite a bit of dependence there, with increasing viscosity causing slower separation of the phases. Given that viscosity actually impacts both the settling speed as well as film drainage, this is pretty much in line with what we would expect in both cases. Okay, so moving on to slag density. Here again, we've got a couple of animations of phase separation from the DMM models, this time at different densities for the light phase. So low slag density results in faster settling and vice versa. with the coalescence of the droplets at the metal pool also probably happening a bit faster at lower densities. Looking at the droplet counts over time, this time it looks like the dependence isn't quite as strong, but it is visible. Increasing the density of the slag phase brings it closer to the density of the metal, which causes slower settling and in turn causes the films between the droplets and the pool to last longer. It's pretty clear that material densities can have a fairly big impact on phase separation as well. And then finally, we also took a look at the effect of interfacial tension between the phases. So this is a really interesting one because it doesn't significantly affect the settling rates of the metal droplets at all, but it does have a very big impact on how fast the droplets coalesce together. The films between the droplets are able to thin out and rupture much faster at higher surface tensions, most likely due to capillary drainage effects, drawing the fluid out faster. And this is really pretty typical in most dispersed phase kind of flow problems. I think it's pretty well understood that one of the ways in which surfactants, for example, affect phase separation behavior is by altering the surface tension. So in the case of molten metals and slags, the interfacial tension is generally fixed by their chemical composition, but some elements such as sulfur, for example, can act as pretty strong surfactants if they're present in the system. Okay, so let me wrap it all up a little bit. Through the course of this project, we've really found that modeling dispersed phase flows is not quite as easy as it looks, especially at this very awkward mesoscale level. In particular, it forced us to dig around under the hood of our favorite CFD software quite a bit more than we really wanted to, but it was a pretty rewarding exercise in the end. We did eventually get things working more or less okay. And what we have now is, well, hopefully anyway, something that will give us a new computational lens for looking at phase mixing and separation problems. Of course, things like this are never really finished, right? We've still got a lot of work to do in terms of optimizing the contact graph handling algorithms. We also need to take a closer look at whether coupled pressure velocity solvers might not be a better idea for these very high viscosity fluids that we're interested in. We also need to think about additional film drainage physics for molten metals and slags, which can have some decidedly weird behavior when you get right down to the micro and nanoscales. But yeah, all of that will keep us busy for a while. I think for us, the main takeaway here was that yes, you definitely still need HPC in your life if you're doing any of this type of multiphysics modeling. It helps you push the boundaries of what's possible. And as we enter this brave new world of AI and machine learning assisted science, working at very large scale to generate the massive data sets that those algorithms are going to need is really going to become the rule rather than the exception. Thanks for listening, everyone. So just before I sign off, some quick acknowledgements. First off, thank you to Mintech, who fund my research and pay me to fiddle around with things like this. And of course, the Center for High Performance Computing, who continue to provide us with access to really world-class facilities and people so that we can actually realize the kind of ideas that we could only dream about in the past. And also thank you, of course, to my co-authors, Oliver Oxterby, Marcus Erwier, and Drian Bezuidenhout. I'm mostly just the front man for the genius of these folks, and I'm very lucky to be able to work with them. So that's it. Thank you very much.
Thank you, Quinn, for the most interesting talk. Of course, we want to know how many cores and nodes you use in a typical run, firstly.
Okay, so typically it varies quite a bit in us. At the moment, the sort of scale of the simulations we're doing, it's topping out at probably a few hundred cores. So we're up at the sort of maybe 10 to 20 node maximum end of things. We've started hitting some quite serious limitations in terms of the algorithm, just in terms of how the graph theory is handled and parallelized, which means we can't really push much beyond that at this point. But the next iteration of the code, we're really hoping we'll be able to seriously stress out CHPC.
Are there any other questions from anyone? I have a question relating to the software process. When you embarked on this, did you have any estimate on how long it would take, and how long did it take in the end?
Yeah, I think you, as scientists, we always tend to underestimate that part of the problem. You know, we kind of see the end solution and we think, oh yeah, I'll just quickly get that done in a weekend. So I don't think it was quite that bad in this particular case, but we certainly knew that we were in for a bit of work. But I'd say we probably thought that the first implementation would take a few months, and it probably did end up taking something like that. What probably caught us a bit by surprise was how long and drawn out the subsequent optimization and code analysis and bug fixing and sort of the successive rounds of optimization that had to come in every time we bumped up a level in scale. I think that was, that was quite surprising. It's, it's definitely not one of these problems that you sort of write up the code and then just forget about it because it runs perfectly everywhere. So I think, that was probably the quick summary of it.
You're not quite complete with the software project, is it?
No, well, as I said, I think these things never actually end. they sort of continue developing. I think there's always a temptation, I think, with us as scientists to say, okay, well, now we've got it working perfectly at 1,000 droplets. What happens? I want to do it at 10,000, and then we'll get it working there. And then it's, what about 100,000? So I think it's definitely, and there will be, I think, some very hard limits that we start to hit with this particular kind of algorithm. I don't think it's endlessly scalable. But
question from Afra Kairala. It says, very interesting talk, well presented too. When connecting the droplets as nodes using the graph theory, do you somehow use their weight or center of mass as a parameter?
Yeah, so we don't have a weighted graph. It's just it's an unweighted graph, an unparameterized graph. It's essentially just saying if you have an edge between two nodes, each node represents a droplet in the collection. And if you have an edge between two nodes, those droplets are potentially in physical contact with each other. So essentially, we have an unweighted graph, which we just need to color. So yeah, that's. But in terms of actually identifying, which the individual droplets and things like that, then you have to take some spatial information into account. Yeah.
And then we have a question from Dr. Charles Crosby. He says, fascinating talk, Quinn. Does any of this translate to more generalization of OpenFOAM capabilities?
Yeah, it's a good question, Charles. I think it's we embarked on this not quite expecting that there was going to be so much sort of software development involved. We've gotten a little bit lazy with a lot of our OpenFOAM development because it does become a little bit like building with Duplo blocks after a while. But certainly this was a project that really pushed us back to looking at the fundamental structure of OpenFOAM and the kind of variables it's got available, the kind of data structures and things like that. And they, it's obviously very, very, very well optimized for the continuum field type problems, but it's and you can make it do these things, but it's as to whether or not it was absolutely the right tool to do this kind of thing in, I'm still not completely convinced. I think now that we've got it here to this point, it makes sense to carry on with developing it here. But it would be, it would certainly be very nice to see some more generalized sort of mathematical structures available, numerical mathematical structures available in OpenFOAM. But at the same time, you also don't want to overgeneralize a tool like that to the point that it becomes a sort of, mostly useless Swiss Army knife that tries to do everything badly. So yeah, I think it's, that can go either way.
We have another question from Dr. Andrew Gill. He says, could the DMM approach possibly be applied to phase change problems like pool boiling?
Yeah, Andrew, absolutely. Anything where you've got generation of bubbles or droplets or things like that, I think a method like this would be very applicable. That's an interesting thought. I hadn't actually even considered the boiling type problems, but that might definitely be some interesting test cases to look at down the line. Thanks.
And then we have a comment from Dr. Werner Jansen van Rensburg. He says, it must be very nice to have an equation named after you.
Yeah, I wish. There was this, unfortunately, this guy called Oswald Reynolds back in the 1800s who pretty much stole my name for everything, the bastard. So, yeah. Good.
Thank you very much for the talk. Okay.
Thanks very much, guys. Thank you for your attention, everyone. Enjoy your coffee this afternoon.
Thank you very much. Bye.
Well, that concludes the session with Ines. We will be starting our session at 3:15. So you're all welcome to enjoy a quick cup of coffee. Thank you, everyone.
Afternoon. I would like to welcome you attendees and speakers to our HPC application breakaway session. We will have two speakers today, and the first speaker is Dr. Rose Mudiba. Dr. Rose Mudiba, I will just read her bio here. Dr. Rose Mudiba is a senior researcher at CSIR Fisher Production Manufacturing. She obtained her PhD titled Computational Modeling Studies of Titanium Platinum Shaped Memory Alloys at the University of Limpopo. Her current research focuses on the alloy development of materials using computing techniques and works closely with experimentalists. She presented her work at both local and international conferences and published several papers. She mentors and supervises honors, master's and PhD students in collaboration with universities. Dr. Mudiba has been actively involved in organizations that support and encourage women in science, technology, engineering, and mathematics. She is also a member of South African Institute of Physics and the current deputy chairperson of Women in Physics in South Africa. So she'll be giving us a presentation on cooperation between computational modeling and experimental characterization of materials. Dr. Rose Mudiba, the platform is yours. Thank you.
Good afternoon, everyone. Thank you so much, Mr. Uncle, for the nice introduction. The presentation will be loaded now.
Thank you.
I've recorded the presentation, so my network is not stable, so we'll play the recording.
Apologies. We can't hear any audio coming out from the recording.
Just give me 1 second. I'm just going to remove my headphones. Just 1 second. Can you hear me?
Yes, I can hear you.
Okay, perfect. I'm just going to mute on my side and restart scene.
Okay. Thank you.
Greetings, everyone. My name is Rose Mujiba from CSIR. I'll give a presentation on the cooperation between the computational modeling and experimental characterization of materials. The outline of my talk, I'll give a brief introduction on the AME, which is the unit that I'm from, talk about the CHPC, results and discussions, conclusions, acknowledgements. We are a unit that falls under the manufacturing cluster at CSIR, which is called the Advanced Materials and Engineering. Our vision, to be the global leader in materials, R&D, and innovation for socio-economic impact. Our mission, we conduct directed research, development, and innovation activities in materials to create, support, and grow the manufacturing industry for the enhancement of South Africans' competitiveness. Our value proposition and focus area. We design, develop, manufacture, and test metallic materials, mill products, and components through powder metallurgy, casting, and fabrication processes. We provide tailor-made innovative solutions for government and industry across the whole value chain of metal extraction to final manufactured products. Now, these are some of our drivers. Our value chain, we have advanced materials, advanced manufacturing, manufacturing services. My focus area for this talk will be on the advanced materials, where I'll focus on the special alloys, on the shape memory alloys specifically. Now, these are some of our project activities and our clients thereof. Now, I'm not going to dwell too much on our technologies. If more information is needed, we can be contacted, and together with my colleagues, we'll take you through all our technologies and products thereof. We have casting technologies, alloys and composites, design analysis and testing, fabrication technologies, powder consolidation technologies, industry business support and services. For this presentation, like I said, this is going to be my focus area on the shape memory alloys. CHPC. It should be noted that on the slide, we only show the activities that we do with the students. However, we do have other projects that the CHPC computing resources are being utilized on. Now, we normally use the Material Studio and media. We access media license through our collaboration with our University of Limpopo, our Materials Modeling Center, which is led by Professor Putin. We have 6 students, of which 1 submitted the thesis this year, 2 are finalizing their write-up, 3 are still busy with the calculations. In terms of the publications for 2020, we have managed to publish 7 papers, and we have submitted some for consideration for publications. Now, most of our students' research work has to do with the focus area thereof is on the titanium beneficiation. Now, I'd like to thank the CHPC for the computing resources. We really appreciate, because this work wouldn't have been possible without their help. So we're so grateful. Thank you so much, CHPC team. Now, results and discussions. I'll be showing the computational modeling and experimental results for the titanium platinum shape memory alloys. The work was done in collaboration with the University of Limpopo. Shape memory alloys are those alloys when a certain pressure or temperature is applied on them, they can remember the previous shape. Now, titanium nickel is one of those alloys that has received extensive application, but it is limited by its transformation temperature to be used as a high temperature shape memory alloy. Titanium platinum is one of the potential shape memory alloys that can be used as a high temperature shape memory alloy because of its martensitic transformation temperature of 1000 degrees Celsius. The structure is known to be the alpha phase at low temperature with an orthorhombic structure. Now, at high temperature is known to be the beta phase with the body-centered cubic phase. Now, studies have been done on this titanium platinum, and it was discovered that the material is unstable with the negative C prime, and experimentally it was found that the strain recovery is less than 11%. Now, in order for us to enhance the mechanical properties of this alloy, a third alloying element was suggested. Now, these are some of the applications wherein we have in aerospace and medical industry. For our project, we hope to use these materials as high temperature shape memory alloys in the aerospace industry. Now, on the slide, I show some results on the titanium nickel, as we'll be using the titanium nickel as our benchmark. Now, the titanium nickel, the B2 structure there is found to be stable at 0K, with all the CIJs being positive. And as you can see, the anisotropy there, it was found to be around 2. Now, with the B2 titanium platinum, the difference is that the CIJs there, our C11 is less than our C12, which resulted in the negative C prime there. So the structure is found to be unstable, unlike the titanium nickel. For the methodology, we used density functional theory within the generalized gradient approximation for our calculations. We used our VASP code. For the convergence test, we used the cutoff energy of 500 electron volts and the K points of 8 by 8 by 8, since they were sufficient to converge our structures. Now, for the properties, would look into the elastic constant of these materials. We have used the B2 titanium platinum, generated the supercell of 16 atoms, and alloyed with different alloying elements on the titanium platinum material. Now, for the results and discussions, on the slide, I show some of the alloys that were generated. As you can see, we have a lot of them. So you can imagine going to the lab and doing trial and error in order to find the suitable material that can be used. So with computational modeling, we're able to save some of the powders, and even the time thereof. Now, for the cubic material to be considered mechanically stable, the B2 structure has to satisfy these conditions. Now, as you can see, like when we add 6% of iridium, our C prime there increases, suggesting that we are stabilizing our titanium platinum with a smaller amount of iridium. When we add more content iridium, our C prime becomes more negative, suggesting that we are destabilizing our titanium platinum. Now, from these, one can say that when smaller amount of iridium is added to our titanium platinum, in terms of the temperature, we are decreasing our transformation temperature. Now, when we add more content of iridium, we are increasing our transformation temperature, since our C prime is more negative as compared to our titanium platinum. Now, this can be understood since our iridium has a high melting temperature. Now, the same can be said with the other alloying element, whereby you can see that for the ruthenium there, when we add a small amount of ruthenium, our C prime there becomes positive, suggesting that the 6% of ruthenium stabilizes the titanium platinum. Now, when more of ruthenium is added, our C prime becomes more negative. Interestingly, when we add tantalum, zirconium, vanadium, hafnium, we see that all our CIJs are positive, suggesting that these alloying materials, they stabilize our titanium platinum. And interestingly, our C prime there, our anisotropy there is found to be closer to that one of titanium nickel. Now, on this slide, we show the results of the C prime versus all the alloying elements. Now, as you can see, there we have our titanium platinum, which is found to be negative, and when we add iridium, ruthenium, chromium, we see that in terms of the temperature, the transformation temperature is increased. Now, when we have our tantalum, zirconium, and vanadium there, we see that when added to our titanium, platinum, they stabilize our structure. And in terms of the temperature, we predict that the temperature thereof is decreased. For the experiments, it was suggested that we alloy on both the platinum side and the titanium side, just to see what happens in terms of the prediction on the transformation temperature of these alloys. Now, as can be seen there, when we alloy on the platinum side, our C prime there is increased, which means our structure there is stabilized. But in terms of the temperature, we predict that the temperature there is decreased. Now, when we alloy on the titanium side with the vanadium, the temperature is increased, but the material thereof is destabilized. Now, for the experimental results, the work was done by Parallelism with the help of Dr. Chikosha and Professor Sia. We can confirm that Parallelism just graduated her master's this year. Now, for the experimental procedure, they have used Pyrometallurgy, using a button arc melting furnace to process some of these alloys. For the microstructures, chemical composition, and crystal structure analysis, they used the SCM, EDS, and XRD. They also did investigate the thermal behavior of these alloys using a differential scanning calorimetry. Now, on this slide, we show the microstructures of the alloys. These are our titanium, platinum, 6%. Then we have our 12% of vanadium on the titanium, platinum. And we have our XRDs thereof and our DSC curves. Now, as you can see, for the pure titanium, platinum, at a high temperature there, we have the cubic peaks there. And when we add our vanadium, we're having new faces emerging there. The same applies with our 12% there. On this one, we show the microstructures for the alloying on the titanium side. And we have the XRDs and our DSC curves there. Now, in order for us to compare our predictions with the experimental, we plotted the multisynthetic transformation temperature with respect to the alloys from the DSC results. Now, we found that when we alloy on the platinum side with our vanadium, indeed our temperature decreased there. And when we alloyed on the titanium side, our temperature there increased in agreement with what we predicted using modeling. In conclusion, we can say that we're able to use both computational modeling and experimental approach to study our titanium, platinum, potential shaping metal alloys. Our predictions were in good agreement. I'd like to acknowledge the following parties. Thank you.
Thank you, Dr. Rose Modiba, for the good presentation that you've given us. Unfortunately, I don't think we'll have time for question and answers, but please feel free to send in your questions. And I know Dr. Rose will be able to give answers to those questions. Thank you. We will now be moving to the next presentation by Dr. Bradley Frank, who is an adjunct lecturer, and he has obtained his PhD at the University of Cape Town. His research interests are in radio astronomy technique, extragalactic astronomy with nearby galaxies, also in data intensive astronomy and big data. Let me quickly check here. For his presentation. His presentation is, he'll be presenting on dealing with the meerkat data deluge, risk for the data center, reward for the scientist. Dr. Bradley Frank, we give you the stage.
Thank you so much, Mr. Chairman. So we're going to play a recording that I submitted a few days ago. So if you could load that up, that would be great.
It will be loaded just now. Thank you.
Hi, everyone. I'm Dr. Bradley Frank. I'm the Associate Director for Astronomy Computing at the Inter-University Institute for Data Intensive Astronomy. My talk is a little different. Here I'm going to explore a few issues that radio astronomers and data center leaders should be aware of if we are to be prepared for the arrival of meerkat data, especially if we want to ensure a smooth road to science. Although much of my work is involved with ILIFU and IDEA, we have successfully shown the CHPC to be a viable resource for radio astronomers. Together, both facilities provide excellent resources for the astronomer on the path to science. And both facilities have already been used to produce publishable science. Last year, we assessed the feasibility of Langau for a mix of radio astronomy use cases. So we did highly concurrent simulations, as illustrated here. We did data management using Globus, and we calculated thousands of meerkat beams for primary beam correction to science data. This image shows how various models or guesses diverge from reality at large radii from the pointing center, but the beams we've calculated are accurate to within a few percent. I would be remiss if I didn't include science images in this talk. This is an image made of the small Magellanic Cloud, unveiling some amazing features around high energy phenomena, along with lots of background AGN. So this is really a testament to meerkat's excellent imaging capability. This image shows a mosaic of 3 adjacent pointings from the MIGHTY survey. The data was calibrated at ILIFU, imaged at Langau, and then mosaicked at ILIFU again. This shows how we're able to move various forms of data between data centers for advanced imaging or processing. Each of these little blips are galaxies detected in neutral hydrogen with meerkat. But the main point of my talk is to present a basic model of ILIFU during meerkat science operations. And It is broadly highly applicable to any data center that is part of the Meerkat value chain, and thus that is part of the SKA Pathfinder value chain. Our simulations provide us with a view of the operational risks, allows us to translate risks to science into risks to the data center, and we can use these models to inform our choices for hardware purchases and integration plans. So this talk provides a high-level overview of an area which we're doing quite a lot of ongoing research. So I just want to remind you of a few important details to set the scene and to outline the problem. Meerkat is a 64-dish interferometer built in the Karoo. There are many large and smaller projects that are planned with Meerkat and will produce exciting and transformational science results. As we observe various fields, the aim is that we have a constant stream of papers and postgraduate projects to ensure that the telescope is scientifically productive. That is, although many of the surveys are multi-year projects and will probably be producing science for at least a decade since their start, we will need to produce regular science outputs. And this places pressure on the data center to keep pace with the observations. So the data scales as the number of dishes, the number of channels, and the number of individual time samples per observations. For Meerkat, the basic observation will include a maximum of 64 dishes. And so for observations that use the 32K mode of the correlator, Meerkat will produce data at a rate of 1 terabyte per hour. And over the course of Meerkat's lifetime, will generate petabytes worth of data. Back in 2009, when science teams wrote their proposals for their large surveys, they put themselves in the big data game. And this pressure for high impact science results means that the data center in turn needs to be responsive, robust, and have enough resources to ensure that we can calibrate the data and allow the domain knowledge experts to analyze their data products. Science teams need to have enough capacity to process their data effectively, but also to diagnose issues and to do quality assurance. So we need a combination of heavyweight compute and highly interactive functionality for science resources. Our model focuses on a very basic time cost equation, and we aim to answer a few very simple questions. How long does it take to transform raw data to high quality science images, for example? And what is the impact on storage? The answer depends on the availability of resources, how these resources are used, and in practice, we will also focus on the data processing deficit, which is the difference in the number of the observations taken and the number of data sets processed. We focus on the operation of a realistic pipeline schematic in which the data traces its way through this workflow to get calibrated. We assume that the model is limited by the most compute or time-intensive step, and at several stages, the raw data is transformed into intermediate data products. We also model the operational schedule of the telescope and the various bits of data transfer and management needed to move the data to the data center. The finite observation time and transfer times are also modeled as part of our model. This is a schematic of our basic model. We assume that each time block is a simple linear function of the observing time. Simply stated, the longer you observe, the longer the transfer and processing time. We also assume that the queue time is independent of the resources requested, and we assume an exponential distribution. The key outputs of the model are the total time taken to process a target data product, and how the storage is affected by intermediate data products. So for example, here, we're illustrating how initially the raw data is dumped onto disk, but over the course of the pipeline, various data products are dumped, that is, they are deleted, and various intermediate data products are written in turn. We assume that our strawman large survey project does 100 observations per year, just to simplify matters. We also cap the total storage to 2 petabytes. We halt computing once the cap is hit, and we start again once we have sufficient headroom. We assume a strawman science case, and we ran the model for a few operational scenarios. We studied the effect on science time if users submitted their jobs to the queue versus having dedicated reservations. Given that new data sets will arrive while previous ones are being processed, we studied the effect of doing concurrent processing. We also looked at the progress of science team by looking at realistic scenarios of observations, that is, with several other projects running on the telescope. This is what the sample results look like. So just to remind you, this is what the model is on the top. On the left, we have the total data volume as a function of time. This is the cumulative plot. The storage increases and decreases as we transform data sets to smaller quantities, but generally increases as we do more observations. We also try to quantify the progress of the science team by measuring how many observations, as indicated in black, have been successfully processed, as indicated in blue. And we assume that we don't reprocess data. The difference between the number of observations and the number of data sets calibrated is called the deficit, and is referred to as the deficit in these plots. In simple terms, the science team and the data center aims to reduce this deficit, as indicated in orange, to reduce this deficit of unprocessed data in as short a period as possible. Consider that each survey will be conducted over several years, and so we will aim to fully process a year or a season's worth of observations before the start of the next season. So as mentioned before, the raw data is constantly transformed into smaller data products. We focus on this deficit of unprocessed data, and again, the aim is to reduce this deficit period so that the project team has cleared a year's worth or a season's worth of data before the next season begins. So this is what the results look like. In this scenario, we have a single queue, but we also study the effect of retaining an intermediate data product called D2 for several periods. So we see the obvious effect of keeping these D2 products for longer periods. There's this large and almost unnecessary requirement for storage that could potentially be detrimental to other users. In this particular arrangement, we see that the deficit time, as indicated here in orange, is more than a year. That is, if we do 100 consecutive observations conducted over 3 months, it takes us a full year to process that data. The main aim of our operations should be to reduce the amount of storage required for intermediate data products as a function of time, and to reduce the deficit time. So we want to move this plot on the left to the lower left, and we want to move the calibrated data curve to the left as well. This can be achieved, science teams and the data center. The science teams need to develop fast and efficient pipelines that produce high quality science images with minimal human interaction and delays. The data team needs to ensure that there is a low queue time, or that the submission of jobs is optimized, or to commit dedicated resources to the survey team. So both the science teams and the data center needs to contribute and collaborate to reducing the hit to storage and the deficit time, thus reducing the risk to science cases. So here we look at the effect of using different queue configurations for a single D2 retention time. So calibrating during one queue, that is one at a time, is the worst case, but providing dedicated resources, a reservation or partition, leads to a dramatic reduction in computing time. So if you calibrate 2 data sets at a time, you get a reasonable reduction. But if you eliminate the queue altogether, you are only limited by the compute time and not by the queue time. So this plot is the same as before, but we dump the intermediate data as soon as possible, leading to a small in-survey footprint of the data storage. So essentially, we want to mitigate the amount of storage that's required by survey teams while they process their data. We looked at a more realistic operation of Meerkat with 2 large survey projects running simultaneously and regular open time observations. We looked at a few more scenarios. So the first and the worst case is where we only calibrate a single data set at a time. And the best case is where 4 data sets are calibrated concurrently using dedicated reservations with no wait time. And we also investigate 2 intermediate scenarios as well. So let's focus on the plot on the right. The black line corresponds to the number of observations, and each color corresponds to each scenario. And there are 2 lines for each color and for each scenario. One is the number of data sets that has been processed, and the other is the deficit. So you see, for the first scenario in blue, you never catch up, and that's because you run out of space. Scenarios 2, 3, and 4 provide you with a chance to catch up. And if you throw enough resources at the problem, that is scenario 4, indicated here in red, you are able to clear the deck. So you're able to process the next year's worth of data. So the point and the main conclusion of this plot is that we can explore various scenarios and try to optimize so we can provide exactly the right amount of storage and compute for science teams so they are able to calibrate their data as effectively as possible. So it's clear here that dumping the data or intermediate data products leads to less storage pressure. So it's less active storage that's required by these science teams as a function of time. And therefore, there's a smaller hitch to other projects on the system. And an increase in resources leads to a smaller deficit time. And keep in mind that increasing more resources also has the effect of mitigating against inefficient pipelines. So initially, you can expect that science teams will not have effective or efficient pipelines, but we can compensate for that by throwing more resources at it. This is obviously quite an expensive way to compensate for poorly written code, but in the case that code is going to be inefficient, there is a scenario to compensate for that. We also looked at, this is the real long-term modeling. So we had the same schematic as we did before, the same scenario as we did before, where we had 2 LSPs and regular open time observations. And here we have 3 scenarios. So in the first scenario, we have, we calibrate 4 data sets concurrently with 2 reservations, and we have automatic purging of D2 data sets. So we delete 50 terabytes per month. So these are what we refer to as the operational scenarios. In scenario B, we have a similar situation where we calibrate 4 data sets concurrently. We have 2 reservations, and we delete data a little bit more aggressively. But we also increase the amount of compute that's available. So we provide extra reservation at the beginning of year 2 and an extra reservation at the beginning of year 3 as well. And we increase the storage by 1 petabyte at the end of year 1. In scenario C, we calibrate 4 data sets concurrently with 4 reservations. We are much more aggressive when it comes to deleting intermediate data. At the end of years 2 and years 3, we increase by 2 reservations respectively, and our storage increased by 1 petabyte at the beginning of year 2. So we start with 4 reservations and we end with 8 reservations. So we effectively, over the course of 3 years, we double the amount of compute that's available to the science teams. So scenario A is the so-called static scenario. So we reach a point at which we are capped, and we can only make a small amount of progress on the amount of data that's coming in. And so here you see that the deficit curve, it does turn over, but it's going to take us ages to reduce the deficit altogether. In scenario B, which is much more favorable, we see that it's not necessarily important to increase more storage. What is quite important is that you are also able to increase the amount of compute. So by increasing the amount of compute, you're able to turn this over, and you're able to calibrate the data at a much faster rate. Scenario C is where you do both. You increase the amount of storage, but you increase the amount of resources available. And here you see that you are able to catch up with the data rate, at quite a good pace. So after 3 years worth of observations, you've caught up with the data within a year, basically. So that means that you're able to serve quite a huge amount of science that would have been produced by these observations. You're able to do that as soon after those observations have completed, as soon after that as possible. So I hope to convey to you that managing Meerkat data is not business as usual, as indicated by these models. Science teams face quite a lot of risk, as I mentioned before. That deficit time means that it takes longer for science teams to stay on top of the data and to produce science. So what we want to do is to reduce that as much as possible. The key outcome is that data centers can take a staged approach to storage availability and also to compute availability. And we need to ensure that there's enough compute for science teams to clear the deck. So we also need close cooperation between science teams and data centers. Good and effective software means that we don't have to throw expensive compute at suboptimal pipelines. So we aim to do continued modeling to ensure efficient operations at the data center, which means better planning and better purchasing. And these models are also important within the broader context of the SKA regional science data centers. So we aim to publish these results within that context. So I'd like to thank the organizers for the opportunity to give this talk. I look forward to any comments or questions that you may have. Thank you.
Would like to thank you, Dr. Bradley Frank, for an interesting presentation. Now is the time for question and answer, or even comments are also welcome. We have, I think is it a question from Queen Reynolds. It says, thank you for your fascinating presentation about the HPC resources optimization. Bradley, did you do any simulation, particularly in the long-term modeling, looking at the impact of unlikely high-risk failure events, for example, partial or complete loss of storage or compute in your pipeline for periods of time?
Hi, well, thanks. That's a fantastic question. So we haven't explicitly included sort of extreme high-risk failures, but our model certainly has the facility to do that. So you've just given me another idea to add a section to our upcoming paper. Thank you very much.
Thank you. We still have time. I think we might be left with 8 minutes of question and answers. So we still welcome questions. If there are more questions, please put them in the question and answer chat. In the absence of any question, we would like to thank Bradley once more for an interesting presentation. I think our session will end here, and the next session will be let me quickly check here. Excuse me for that. Our next session will be at 4:30. It will be a keynote presentation. Thank you all for attending, and we would like to thank all our presenters for giving interesting presentations. The session has ended. Thank you. Back to the the program manager.
Okay.
I'm Kwe. Thank you so much to everybody who attended the session. Our apologies for the technical glitches we had earlier in the session, but I'm sure everything went well and the talks were very interesting. Please do sign in or join us for the keynote 4 that is taking place at 16:30. Thank you very much. Have a great day. Bye-bye.
Thank you. Bye.
Bye-bye.